So I suspect that for those who've been analysing all of these large data sets or managing our ability to look at them, what they really want to do now is have a bit of a break. But I would like to ask what you think the next major step will be from these data sets. What's exciting you most about what we might see? And I'll start off with asking that to Robert.
Hopefully I gave a hint to that in what I presented in the talk. I think clearly novel insights from the genomes alone, but I think one of the things I'm really excited about is the integration of the data from the genomes, from the proteomes, from the methylomes, which I'm keen to hear more on, from the deep phenotypic data, from the primary care data, for which we heard such a cogent argument earlier. I think through the integration of those data, there's huge insights to be drawn at that. I hinted at a couple of examples of that.
Really, Robert, what's left after that? You had to take everything? I think one of the things I'm also really excited about, and again, this goes back to the talk that Julia gave, I'm really excited to see what we can learn with all of this now that we're integrating all of this data, thinking about things like risk prediction as well.
I'm really interested to see what everyone does with this data, how we can make all the analyses which are being done really reproducible, how we can ensure that it's really open, the results that everyone's getting, and that every researcher can then build on the steps of what's gone before. Working at these huge data sets, we don't want everyone starting from zero. We want everyone starting from the level that was reached previously. And that's, I think, a huge challenge.
Yeah, I mean, I'd echo the comments about integration. I mean, I think this is a really unprecedented opportunity to put, multiple layers of genomic and other types of data together in a way that's not been done before in really large scale. And then, look back at, what the predictors are, but also use the kind of measures that are being generated and we hopefully will plan to generate to predict forwards as well and develop really robust molecular predictors of risk and how we may mitigate those issues.
I think for me, in terms of the analysis of the 500,000 genomes and the many different things that we could do there, the thing I'm most excited about is getting a better understanding of the role of rare non-coding variants in human health and disease. We know that they play a role, and there, up until now, hasn't really been an available data set to systematically understand the extent and the breadth of that role. Of course, the challenges in understanding those variants and identifying the subset that actually have some functional impact. And here the UK Biobank is super valuable because, as Dr. Miller explained, we can use things like the proteomic data to actually help us to identify those variants that are having an impact, both coding and in the non-coding space. And in terms of the impact on drug discovery, it allows us to start to think about things like non-coding targets, which is really exciting. So I think that would be the area that I'm most looking forward to exploring.
Fabulous, thank you. And to pick up on a question that's come in from the online audience, they are asking whether there are any efforts to characterize omics across tissues and to look at tissue specificity. And perhaps I can add to that and say, is there anything else you think we're missing? Clearly, methylation across the entire cohort would be fabulous. But is there anything else you think that we need?
Jill, I'm happy to start. So obviously, it would be brilliant if there was a mechanism by which other tissues could be collected eventually. I think it came up in it was touched upon in the questions in the dementia session this morning. Obviously, I'm biased. I'm very interested in the brain, but to me, this is a kind of once in a one-off opportunity to have all these measures that we've got now in peripheral samples, and then if there was a way of embedding post-mortem biobanking into UK Biobank, I think that would be game-changing with the ability to then potentially predict from a measure that you've taken peripherally about variation in something like the brain that obviously you can't access in living individuals. So I think that would be a brilliant opportunity going forwards.
I'm not aware of any major drivers, and perhaps Rory or Lucy could comment on the wish to sample from diverse tissues from UK Biobank participants. I'm sure there are participants in the audience. I'm not sure that that's imminent, but I think it's highlighted there's growing interest and, in fact, demonstration in recent days, high-profile, really exciting papers that demonstrate that from the really valuable data from plasma that's been generated, there's growing ability to characterize tissue and organ-specific disease processes. So I think there's huge opportunity there in UK Biobank that certainly we are thinking about. I'm sure many others are too.
I think the other thing I would add, which will be challenging, but maybe slightly less challenging than tissue, is longitudinal data. We are going to have a cohort of 100,000 people, most of which, or 70,000 of which, will be repeat image. So we'll have 3 data points on people. So changes in the methylome, changes in the proteome. So I think the longitudinal aspect is also something that's going to be really important.
Thank you. Okay, I shall open it up to the floor if anybody has any questions. There's one at the front here.
Thanks. Hi, Alex from the University of Cambridge. So this is a question, I think, primarily to Jonathan, but I think also to the people from pharma. So one idea I've seen in the literature with the methylation clocks now, for example, is that one could use these as biomarkers also in clinical trials in cases where we have an outcome where currently, I don't know, we have to wait 10 years until someone really develops a certain kind of disease. And I always wondered, there's regulation and clinical validation and so on. How close are we to this? How realistic are these things? Because I've seen a lot there, which seems cool, but maybe a bit science fiction-y. I was wondering whether you could comment.
Well, I think discovery in the cohort like UK Biobank would actually get us potentially towards that stage. So, I showed the smoking example, and that really is, it's a near perfect sort of biomarker of exposure. And the clocks, already really robustly predict actual age, and there is some evidence that they are, really indexing accelerating age. So if that's true, that opens up the potential of monitoring those things in clinical populations, kind of real time, really easily and cheaply, because it's quite easy to get a blood sample and kind of directly measure those. So I think it's a really exciting question and idea. Yeah.
I think there was another question over here, number one.
Yeah, hi. My name is Elias. I'm a researcher at Weill Cornell Medicine in New York. I'm curious again for Jonathan, but anyone else could also jump in about what do you think the relative benefit of methylome versus proteomics is for exactly these kinds of more sort of physiological, episodic information that may change over the course of a lifetime of an individual versus, just specifically those two, but also there's metabolomics, just many different omics you could look into. Just wanted to get your thoughts on the relative value of those things.
I'll let you start.
I think I'm really going to hedge on this one. I honestly think the relative value probably does depend on your question. I think that we would see cases where perhaps the methylome is way more sensitive in a specific disease, but where the proteome is more sensitive in another disease, or where one of them is going to be able to, maybe the proteome does better with things that are acute, whereas the methylome is maybe better for things that are chronic.
And I guess the proteome is sort of closer to that endpoint in terms of what's going on in the cell or the body. Well, I think the real exciting potential here is sort of training these methylome predictors against the protein measures that have been developed. And I know Riccardo Marioni's here. He's done some great work on that in his research, and extending that into Biobank would be just fantastic, I think.
Thank you. Are there more questions? Yes, there's one over there. Number one, please.
Hi. So, I mean, obviously, it was super exciting. Question maybe for people from the, Lucy from the UK Biobank. And you touched upon this in one of your slides about the integration of different WGS databases. And you showed an example. But maybe I can go a bit beyond that. So maybe not so much now, but in 5 to 10 years from now, when there'll be a lot of different WGS databases, I can imagine there being a pretty significant analytical challenge in the integration. So for now, so what we're interested in is a part of the genome that can only be looked at using whole genome sequencing. So we've accessed the UK Biobank and we're hoping to access all of us. Now, to run all our analysis and all of us, essentially reinventing the wheel. It's not just which aligners use, it's pretty much everything. Now, maybe 5, 10 years from now, if there was a single portal where we could access different WGS data sets that have hopefully been processed in a similar way and have been harmonized, it would make things so much easier. Now, I can imagine so many challenges with that, dealing with ethics and everything else from different countries. But I can see this being a very significant challenge 10 years from now, and maybe it's something to think about. That's,
Yeah, it's an interesting point. I think, obviously there's vast numbers of data sets out there. I think meta-analysis is a really promising approach, particularly when you have different data sets which have been processed in different ways. Aligning on different ways of processing data sets in the first place is also looking like something which could be a really positive step forward. I think the other challenge will simply be the size. Our data set sits at 30 petabytes. If you bring in 5 more data sets at a similar size, computing on that is also going to be, in one big go, is also going to be a massive challenge. I think there's a lot to keep the scientific community busy over the next 5 to 10 years in terms of that as well.
I might just add on that. I think algorithms approaches will improve and standardize over 10 years. I love the notion, if I could vote for that to happen, of data standardization and collation into one place. I like that. One of the comments I make is that even in the UK, standardization, centralization of data has proven really challenging. So 10 years feels great, perhaps optimistic, but let's see what we can do. Even in the UK as a start would be great.
Brilliant. Thank you ever so much. There's a question over there, number 2.
Hi, Arun Hingorani from UCL. Thank you all for the wonderful presentations. My question's to the pharmaceutical company representatives. With the increasing use of genetic insights and genetic target validation for prioritizing pharmaceutical research and development using resources like UK Biobank, are we likely to expect recognition of public contribution to creation of value in the development of new drugs in the form of drug pricing and better access to drugs in the future? How's the economics flipping, and what should society and the public expect to see in the future? Thanks.
As all of the pharmaceutical companies back slowly into the shrubs.
No one's very keen to take that. Keren, you were about to go.
Well, I'm not sure I have a specific answer for you. I'm afraid it's rather an enormous question and rather outside my remit, but I absolutely accept the point. And drug discovery and development is an enormously complicated and difficult and expensive endeavor, and appears sometimes to get increasingly so as the low-hanging fruits are taken. But nevertheless, in all kinds of ways, I think that at the research and development side, acknowledging the contribution of the participants, the funders, and everybody else who has made these amazing data sets happen is absolutely vital.
Thank you for those answers. I think that's definitely one of those questions which we can say we will continue to discuss over drinks later on. One more here, number 3.
Yeah, Mike Pietzner from the Berlin Institute of Health. I want to pick up actually on the question before. I think this data access question is more imminent than 10 years because, I mean, UK Biobank is great, but it's a sample from the population, and for many diseases, we're simply underpowered, in particular if you want to go for rare variant discovery. So what are the more like plans within 1 year, 2 years to get data? I mean, you pull data from Genomics England and all of us and so on in the same analysis environment. So why not making this available now, or at least allowing us to download some of the data for whole genome sequencing? Not to say all 30 petabytes, but at least the called variants.
So, yeah, I mean, it's something which is an important consideration. I think the data which is processed using Dragon, it's not processed together. It's using the same pipeline, the same approach. So although there's comparative processing and comparative resultant data sets, these weren't pulled together for analysis. So, you know, there isn't a unified data set that is sort of not accessible. They are still separate data sets, despite having been done on similar approaches. I think it's something we have to keep considering. I think the cloud model works really well, and it provides an enormous amount of opportunity. Many people would not be able to, I think probably most people, download and process on premises the data from one of these data sets, let alone all of them. And I think establishing the best ways to analyze these sort of separately and combine results, and statistically how to do that, is a real challenge, but an important one, and perhaps sort of the best first steps.
Maybe I can just add to that. At AstraZeneca, the way that we've been thinking about this is exactly as Lucy previously said, which is running analysis separately on separate data sets where they have been processed separately, and then sort of doing a meta-analysis to try and combine them. And there's probably a lot of room in terms of methods development, in terms of the best way to do that and how closely can we align the upstream pipelines. But in terms of sort of a realistic way of getting short-term access to the data, that's how we've been thinking about it.
Thank you. Thank you ever so much for your answers. Thank you for your questions. As someone who can't even keep my desktop organized, I'm grateful to people who can manage 30 petabytes of data. So perhaps you would join me in thanking our speakers from this session.